Hello, and welcome to this brief tutorial on how to use some of the features at the NCBI website. Let's get started. First of all, please enter NCBI into your Google browser. There we can see the very first link here is the National Center for Biotechnology Information, or NCBI. So what I'd like you to do is to click that link. And this will bring us to the key NCBI websites. This is where all the magic happens. So now that we're on the website, I would recommend after this video is over that you should take some time to explore all the various features that we're going to be talking about. But just to give you a brief overview, I'd like to point out a couple of things. To start with, the how-to links are right down in this region of the screen. And as you might be able to guess, the how-to links are going to show you how to accomplish a variety of specific tasks that you can do here at NCBI. So I'm going to click that link there, and you can see it takes us to this screen. And there's a large number of different how-tos that you can find here. You may have to use one or more of these with help of your particular project. Some of them may not apply to you. For example, submitting data to NCBI, perhaps not right now. But you certainly may want to find gene expression patterns. You might want to view the 3D structure of a protein. You may want to learn how to run BLAST software, and especially down here, you may want to be able to complete the NCBI tutorial, so you can learn a little bit more about some of the other features that we won't have time to talk to you today about in this video. So now what I'm going to do is I'm going to scroll up here, and I'm going to show you another interesting place of resources that you can go to, and that is the databases. So if we click right here, there's a tab here that says databases, and these databases are essentially all these different compiled resources that NCBI has and that are all searchable. So you can find whatever information you need from these databases. I'm not going to have time to talk to you about all of them today, but a couple of ones that we're going to point out here is, for example, the bookshelf, which I'll show you how to use in just a moment. It's sort of a repository of a lot of biomedical textbooks. There are also things like the conserved domain database. These will tell you if the sequence in question that you're plugging into NCBI has protein domains in it. What else do we have here? GenBank here is a United States-based sequence repository. We have a lot of different sequences that are stored there, and of course other things like Gene, which we'll be using later too. And Gene will let you basically search and find information about particular genes that are of interest. So let's scroll back up for now. I encourage you to explore more of these yourself. We're going to scroll back up. You could also find these different databases, pretty much the same ones, by clicking up here. Notice how it says all databases by default. If we press down, we can enter a search term for a very specific database, for example, PubMed. PubMed is a citation database for biomedical literature. You can also search in something called PubMed Central, and that is essentially a PDF storage site that will store the actual PDFs associated with primary literature articles instead of just the citations. So they're similar but a little bit different. Protein, nucleotide, genome stuff. You can look at books, which we'll go to in a minute. Genes, all that stuff that you might be interested in is going to be somewhere on this list. So that is that. Now let's hit the back button on our browser and go back to the initial home page. You also can just simply hit this NCBI link up here in the upper left, but I'm going to choose to go back a couple of times. And now we're back on the home page. So what I'd like to point out here are the popular resources. These are resources here on the right-hand side. And as you can guess by the title, these are things that are very, very commonly used. For example, PubMed, Bookshelf, PubMed Central, PubMed Health, BLAST. You can actually see I have used these. That's why they're in purple right now. As well as a bunch of other resources over there. There are some interesting features down here. This will kind of scroll back and forth as we are working. Well, one thing I want you to be aware of here is number 2 is that NCBI has its own YouTube channel. So if you like to watch videos that are related to NCBI, you can go to YouTube and find them there. There's a lot of different tutorials that will help you. For now though, let's get started actually doing something. How about we go into the bookshelf area, click right over here where it says bookshelf on the right. This is equivalent, just for your information, of going to all databases and scrolling down to where it says books. That's essentially the same way, but we're going to just go over here and click the bookshelf button, as I said. And what this allows you to do is to search a large number of biomedical books and other books for a particular topic. So for example, if you just happen to be interested in something like dengue fever, type that in there, hit enter, and you can see there's a large number of books here that come up. And as you can guess, these have names that would lead you to believe that they probably have some information about dengue fever in them. For example, the global burden of diseases and risk factors, disease control priorities, infectious disease. If we scroll down a little bit more, there's this particular one I want to show you here, this Medical Microbiology 4th edition. Now, what I want you to be aware of is in order to actually see where dengue is listed in this book, you don't want to click this link here. So this top link, I know you're going to want to click this link if you've never done this before. This will just take you to information about this book in particular, this Medical Microbiology 4th edition. What you want to do is go to the lower portion of the screen here and either say see the top results of this book or see all results. I'll just click see the top results right there. We'll see what happens. You can see it brings up a variety of chapters here that might relate to dengue fever. Here's one on viruses, so I'm going to click that one there. And this essentially is the chapter. So one thing I'd like you to remember to do on a lot of NCBI websites, it's not always obvious, but if you look over here on the right at the slider, you can see you can actually scroll quite a long ways down. So just to give you a quick example here, hopefully this doesn't give you motion sickness, but there's actually quite a bit of information, a whole chapter's worth of text on this web page that you can find if you're interested. But our question was, does it have anything about dengue fever? I'm going to hit Control F. It's a keyboard shortcut on Windows machines. If you have a Mac machine, you can hit, I believe, Command F, which should do the same thing. Then I'm going to hit dengue right here. And if you see, if I keep hitting the next button, dengue is actually listed a large number of times in this document. So there's quite a bit of information here to be had in the bookshelf. That's kind of our brief demo of the bookshelf. Let's go back by hitting the back button a few times to get back to the home page. And what I'd like us to do now is to use the BLAST tool. BLAST is one of the most important tools in NCBI for molecular biology purposes. I can personally say that it's probably one of the most common things that I use in NCBI. Probably the most common, except for perhaps maybe PubMed itself. I'm going to click on the BLAST link right there. And this will take you to the BLAST page, and you can see BLAST stands for Basic Local Alignment Search Tool, which is not a very exciting name, which is one reason they call it BLAST, which sounds much more interesting. And over here you can see there's actually a large number of different options of things you can click on. They're always designing new different types of searches. But basically what BLAST is it's going to allow you to take an input sequence, say a nucleotide sequence, and compare that to a database to see if anything similar has been found. And it's a great way to identify an unknown sequence. If you have an unknown sequence you want to identify, or if you have a sequence that is known but you want to see how closely it compares to, say, maybe other groups of organisms, BLAST is a great way to do that. So 9 times out of 10 you're going to want to use this over here called Nucleotide BLAST. There's a large number of options here, but we're just going to talk about this first one, this Nucleotide BLAST today, and it's probably the most commonly used feature of BLAST. I'm going to just click there, and the screen here gives you a lot of different options that you can control, but we're only going to mess with a couple of them, so don't freak out if this is an alarming number of boxes to fill in. The first one is we have to enter a query sequence. That goes right here. And the query sequence is essentially the sequence that we are going to input and search for in our database of our choice. So I happen to have a query sequence somewhere over here. Here it is. I have mystery sequence in the bottom. And we have our mystery sequence of nucleotides. Who knows? This could be anything. Apparently it's called sequence 2. What I'm going to do is I'm just going to search, select a bunch of this. You might have your own particular sequence of interest. Any sequence will essentially work, but you'll get a little bit different results. I'm going to paste that in over here. And then I actually, there's a couple options here. If I happen to know this is human DNA, which I do know it is, and I was only interested in looking at humans, I could look at this Human Genomic Plus transcript database, but I'm actually going to choose to use the nucleotide collection, which essentially is blasting against pretty much all databases, and what it'll do is we'll be able to see results from other species. It just gives us a few more things to look at. So really, this down here, and then the query is pretty much all I ever mess with. I don't really touch any of these other options, although they are there for you if you ever needed to use them in the future. Now I'm going to press the blast button down here in the lower left-hand corner. So click that. And this is usually relatively quick. Sometimes it takes a few seconds. Oh, there it goes. Great. You can see at the top there's a lot of information about our sequence that we entered, including that it's about 2,975 letters long, or in this case nucleotides long. It tells you that it is a nucleic acid. It's identified it as one. Plus, I think that's how we entered it in. It tells you what database you were searching. And then just below that we see some pictures. Now, before I get to this stuff down here, I'd like to mention that for some proteins that have very conserved domains, nowadays, like BLAST will actually show you the conserved domain sort of up here, usually between this blue box and this lower region. So if you see that, that's actually another great tool. We're not going to talk about it today, but it's kind of a neat resource where you can find and localize domains in your particular protein. So down here are a whole bunch of red lines, a bunch of stripes, and these stripes represent the various sequences that NCBI has found, or BLAST has found, in a nucleotide database. These are sequences that are very similar to our query sequence, and you can tell by the length of the line how much sequence the matching sequences, or excuse me, let me say that again, how much the sequences that are found line up with the sequence, your query sequence. So on top here you see our query sequence goes from 1 to almost 3,000 base pairs over here, which if we remember was about how long it was over there. And these top ones line up with essentially that whole region. The ones a little bit lower than that, you can see they don't line up quite as far. They're kind of missing the beginning. And these ones over here line up even a little bit less. So there are a few different variables that we're seeing there. Now, if you highlight over one of these, you can actually see a lot of things in NCBI. You can just highlight over, and it will quickly tell you what that is. This here says it's Homo sapiens beta globin region of HBB gene. It's a hemoglobin gene, and I have to move the mouse cursor, but it says that right over here in this particular zone. So if I mouse over, you can see that there. So at this particular time, what I would like to ask you, and a little bit of a quiz here, is why is there more than one match? We searched for one gene. We've discovered what that is now. It's this HBB gene. Humans essentially only have one of these genes. Why is there so many things in this list? So I'll give you a second to think about that. Great, yeah. So the answer to that is that there's actually a few different answers. One is that multiple people did experiments. Some people sequenced the whole genome. Some people sequenced maybe different forms of mutant hemoglobin from people that have blood diseases or have mutated hemoglobin molecules. And also some of this is actually going to be from other species. So if we look carefully, let's see if we can find one. Probably one of the lower ones down here. Oops, can't go too low or can't see it. These are all different Homo sapiens isolates. If you're scrolling over and following along at home, you might see something that says like pan troglodytes. That would be chimpanzee. There's sometimes you see G. gorilla in here for gorilla. There's an orangutan one in here. I'm not really seeing them right now, but they are actually in here. Depending on how the search is done and how much sequence you've used. If you use less sequence, you actually get probably more hits to unrelated things like other primates. Now I'll tell you, this time I actually chose HBB to use just because it happens to be a very particularly small gene. It actually is rare in the sense that it fits on a page of one piece of paper. Normally genes would take up volumes in some cases. I also was chosen because HBB is a very highly studied sequence, so that makes it a little bit easier and certainly gives us a lot more hits for this purpose. What I didn't tell you is the color of these bands tells you how closely they match our query sequence. So red is very good. You might see red or pink. I've actually never seen these other colors, the black, blue, and green, but if you're sequencing something that's not very well conserved, you may see those. Let's scroll down a bit. We can see all our hits are also listed down here. The same things in this table are exactly the same as the red bars listed above. if you were to compare them. This first one here is exactly the one I read to you. And there's a lot of information down here, so let's kind of break it down. First of all, you have the accession number. This is essentially kind of like a barcode number, like you might see at like a library, just to keep track of something. If you happen to know that number, it's like a shortcut to find out where this gene is. I never worry about them too much. You also have a couple of scores here, the max score and the total score. I would recommend essentially ignoring those, at least for now. They do have a meaning, but it's not important to us at this time. The query coverage tells you how much of our sequence matches the sequence that was found in the database. So in this case, 100% from the very first nucleotide to 3,000 bases out all line up with this particular gene. You can see some of the other ones only 97% matches over there. Now the e-value is essentially telling you how good of a match what you found is. It's essentially a measure of how likely something is to occur by chance. Because as you know, when you only have 4 different nucleotides, there's a certain probability that you'll just coincidentally get something to line up with your particular sequence of interest, especially if it's short. We have a very long sequence, so our e-value is essentially 0 or very near 0. And that's actually very good. So that means there's essentially no possibility this occurs by chance. Common e-values are things like e to the negative 23rd power. That's a very small number. If you ever had an e-value that was large, like say 1 or 5, you'd want to be skeptical if that maybe is not a very close hit, or there might be some random chance going on. The other thing to pay attention to here is the max identity. And this is important. This tells you how many of the nucleotides in our sequence that we found over here match exactly with what was in our query sequence. So in this case, it's a 100% perfect match, because the sequence that I used, this mystery sequence, was actually probably taken right from this particular site. So it's a 100% match. It covers it exactly. You can see some of the other ones a little bit lower. They're not perfect. They're 99%. They maybe have a couple of nucleotide changes, and that's as you might expect in different human haplotypes of the beta globin gene, and there's quite a large number of these here. So if we scroll down a little bit more, what I'd also like to show you, if I may, if we just look at our first one, you can actually see the lineup, the alignment it's called, of your query sequence, the human, or actually our mystery sequence here on top, and the subject sequence down here is that human HBB gene that we isolated. And you can see all these lines indicate that it's a perfect match. If it's not a perfect match, some of these lines will be missing to signify that there's a mismatch at those locations. And that can help you identify particular sequences. So, pretty interesting thing to do. So now, what I'd like to do is, now that we have identified our HBB, or excuse me, now that we have identified the mystery sequence as HBB, let's go back to NCBI and try to find out what we can about this particular gene. So I'm going to hit the back button a couple of times, and if you want, you can go directly to NCBI if you prefer. I'm going to go to the gene option down here. See the gene. Click that. I'm going to type in HBB. Hit enter. And there it is, the very first one, HBB human hemoglobin, is located right there. Over here on the right-hand side, you can see a lot of other organisms that also happen to have hemoglobin. There's musculus for mice, Homo sapiens for humans, Bostoris for cows, Rattus, I can assume you probably can figure out what that is. Pan-troglodytes is chimpanzee, and there, of course, are a number of others there as well. But for now, let's just click on this human one from Homo sapiens, the very first link. And we'll scroll down a bit. You can see there's a lot of information about the gene located in here. And here's sort of the genomic context of our gene. If we look, our gene is listed right here, HBB, and it has an arrow. The arrow here is indicating the direction of what? So the direction of the non-template strand, 5' to 3', as the direction the mRNA that would be created would be made in. This is a piece of chromosome 11. These other things, or 5'2", Z1, HBBP1, or other nearby genes that happen to be close to HBB, or HBB, excuse me. One question I'd like to ask you now, let's do another little quiz, if you don't mind. Why, well, actually, before we do the quiz, let's find out what these other genes are. If you mouse over things, remember, you can actually oftentimes find out what they are. So we know the one on the left here is human hemoglobin beta. HBD is hemoglobin delta, and HBBP1 is hemoglobin beta pseudogene 1. So these are all very similar genes to each other. They're all related. Quiz is, why might these three things all be close together on the same chromosome like this? So think about that for a minute. Okay, I'm sure you did very well on that little quiz. And yet the answer actually is that it's most likely caused by an ancient mistake of sorts. Polymerase will occasionally make mistakes. You make mistakes probably all the time. I know certainly I do. Most of the ones I make don't have repercussions that last millions of years. But in this case, an accidental copying of some original precursor hemoglobin gene ended up creating these different derivatives. Here in HBB that ended up becoming broken over evolutionary time, and a different one here, this HBD, which underwent divergent evolution and actually had some different things happen to it over there. So let's scroll down a little bit, if you don't mind. Here we can see in this genomic regions, transcripts, and products page, we have this green box right here. It has a label. You can see labels right there. And if you select anything with your mouse, you can see what it is. So I'm going to actually click this green thing on the top. You can see there's the green thing here. The next line below that is blue. And the line below that is red. And if you look carefully, it'll actually tell you right here what those various colors represent. So, quick quiz. What do these colors represent? What do the green, blue, red, and black colors represent? So, hopefully this should be a pretty easy quiz for you. Great. Alright, so you figured it out. You figured out that this is the green is the gene, blue is the mRNA, the red here is the protein, and these black ones here are exons. They show you where the exons are located. The light green color here at the very 5' end and the very 3' end, those are the untranslated regions that are present. The straight lines are the introns, and the boxes are the exons. They correspond to these black exons down here, as you can see. And if you look carefully, you can see essentially where the protein begins, right about here. This is where the start codon is going to be on the mRNA, and where the protein ends over here, if we kind of go up to the blue line, that's essentially where the stop codon is going to be. So a lot of interesting things there. Now, a good question for you here is, look, on the top, HBB, the gene, was pointing to the left. And down here, it is pointing to the right. So the question is, why is that? So think about that for a second. The answer, of course, is that HBB happens to be in this direction on the chromosome, according to convention. But to make it easier to read, just by convention, we typically like to write things in the 5' to 3' direction as it would appear on the mRNA. So the computer and NCBI has automatically put it in this orientation to make it a little bit easier to read. It's a very handy feature that I happen to like about it. Now, some students get a little bit stuck at this point, and they start thinking that on your chromosome, the top strand or the bottom strand really matters. The top and bottom strand don't really matter. That just depends on which way the helix is turned at that period of time, about its axis. The top and bottom strand doesn't matter. What does matter more is the relative positions of the genes and the 5' to 3' direction of those things. Next, what I'd like you to do is to go up here where it says go to nucleotide graphics and press this button right here that says graphics. And if we click that, what we can see, there it is the title at the top now is Homo sapiens chromosome 11. So we're looking at a whole chromosome, and you can tell that because these numbers are listed in megabases, 20 megabases, it's like a million base pairs, all the way from 1 down to the end. And these little green things here along the way are genes that are present on the chromosome. This blue line indicates where our particular gene of interest is, HBB. Right here, it lies right there. If you want to, you can easily scroll in either direction just by clicking and sort of dragging, like so. You can also kind of zoom in and out, like that, or if I go the other way, you can make HBB very small. If you click this plus ATG, you can go all the way to the nucleotide sequence, which is kind of neat. You can see the exact sequence at any spot and scroll along. Why don't we go back to where we were? The back button, please. And this is actually a very similar diagram. It just doesn't have that whole chromosome region that the last one did. Go to the Tools section, and in here go to this section where it says Sequence to Text View. This is very important. Don't forget this particular step because it's actually a great way to visualize things, in my opinion. And at first it looks like we have just plain sequence, but if we scroll down, you can see there actually are colors associated with the sequence. So another good question for you might be, what do these colors mean? So let's take a little quiz about these colors and about what they mean, and see if you can come up with some of the right answers to this quiz. Great! Hopefully you did very well on that quiz. I'm sure you all did great. I want to thank you very much for following along with this particular video demonstration. If you have more questions, you can feel free to watch the video again or go to the NCBI tutorial as I showed you a little bit earlier in the video. But for now, I recommend that you go find your own gene and learn about whatever you can from NCBI. How many introns are there? How many exons are there? How big is the gene? What chromosome is it on? It's up to you. Just find any gene that you're interested in and practice out these skills, because I have no doubt they will come in handy on your future. So with that, I will say happy exploring and wish you good luck, and hope you have a good day.